for having me here. I'm excited to give this talk and describe some of my work on designing facilitated dissociation. So I guess as Lukman said, please feel free to interrupt me at any time for questions. And Yeah, let's get started. Okay, so I wanted to give first a just a brief overview of kind of the general like workflow for thinking about de novo protein design. So compared to like the central dogma of biology, um, where information is encoded in DNA and then it's transcribed to RNA and then translated to proteins, and the proteins have some function. De novo protein design kind of inverts that. So rather than starting from DNA, instead we start from some desired function that we want. Uh, then we can generate structures which should perform that function given the like structure function correspondence in proteins. And then we can generate sequences with amino acid sequences, which should fold into those protein structures that we designed. We can also use computational methods like AlphaFold to check whether the sequences that we generate are indeed predicted to fold into the structures that should perform the functions that we designed. And this is one of our main ways we computationally filter designs before testing them. Designs which pass those filters, we can uh, we can reverse translate that amino acid sequence into a DNA sequence. And then here's where we can go from, the world of computers to actually the real world. And we can synthesize the actual DNA molecules encoding our proteins, put those genes into real cells. They will express our protein from those genes, and then we can purify that protein and measure whether it actually performs the function that we designed. And if that matches, then that's a successful design. We can also go a little further and using experimental structure determined methods like crystallography, we can show that uh, in addition to the function matching what we designed, we could also show that the actual structure of the protein we designed matches the structure we designed it to fold into. And then that shows not only does this protein perform the function that we designed, but also it performs it for the reason that we designed it to. And so our structures are like our structural hypothesis for the sort of structure that's required to perform the function is indeed correct. Okay, so now I'll talk about the problem I actually wanted to solve and the function that I was trying to design. So generally, proteins need to interact with some target in order to perform their function. Uh, Proteins very rarely act in isolation, rather they're always performing some action on other biomolecules. And so binding is almost a prerequisite for almost any function. Uh, High affinity binding is uh, useful because it often leads to more downstream effects. So high affinity binding here between what I'm going to call throughout the rest of the talk a target in pink and a host in blue. Uh, Binding is high affinity when generally when the off rate is low. So there's a very low um, rate of dissociation of that complex. That means that those complexes are going to remain bound for a long time. So there's a really long lifetime for those complexes. And just in terms of energy landscapes, high affinity binding tends to look like um, there's both a high energy difference between the bound and unbound states, and also there's a high barrier for the dissociation process. So this height here is quite high. Now, if you want to make the system a little more complicated and now have a way to turn off a high affinity interaction, generally you would do that through direct competition. So you add this third component, which I call the effector, and then whenever the target dissociates, the effector can bind in its place, and then that displaces the target. But the overall rate of this competition process is going to be slow when the target host affinity is high, because that target off rate is slow. So that like this energy barrier here for target dissociation limits the rate of the overall process of competition. So we were kind of inspired by DNA, which specifically this phenomenon called toehold-mediated strand displacement, which actually lets you have both high affinity interactions that can also be rapidly outcompeted. So how does this work? So here there are two single-stranded, it's like a DNA complex, a complex between two single strands of DNA, where one is longer than the other. So there's this toehold here. And then when you add a fully complementary strand, it can kind of weakly associate at this toehold. And then through this process of branch migration, it can quickly outcompete the strand that was previously bound. So it leads to its rapid dissociation. And this can speed up the rate of dissociation of this red strand by like millions fold. It's really crazy. So also this behavior exists in proteins and is useful in biology for certain things. So NF-kappa B, for example, it's involved in the stress response. One protein that it expresses actually is its own inhibitor, I-kappa B, which can bind to, so it's this protein here in light blue. It binds to the NF-kappa B and actually rapidly displaces it from the DNA. And then that turns back off the stress response or the NF-kappa B signaling so that it limits like wasteful overexpression of genes. And then also in motor proteins, like here I'm showing dynein, uh, ATP can bind and induce conformational changes in the motor, which lead to its rapid dissociation from the track. And then this helps this like induced dissociation helps coordinate the overall mechanochemical cycle of dynein. So we wanted to design a way for this effector protein to, instead of having to wait for the target protein to dissociate spontaneously, instead this effector could directly bind and induce the dissociation of the target. So how would that work? So we designed host proteins, which now have two binding sites, one for the target and then a second one for the effector, where the effector could bind. And also with two conformations, one which binds the target strongly, and then the other induced by the effector, which binds the target more weakly, leading to its rapid dissociation in the presence of the effector. So then now, in terms of energy landscapes, we have this kind of strained, excited ternary intermediate along the pathway, which overall should have a lower energy than everything being fully dissociated in the mutually exclusive competition case, so that overall the energy barriers along this new designed pathway could be lower. And then, so to actually design this, we first wanted to design this strange ternary complex, where binding the target and effector are allosterically coupled. And second, we needed to design like a rapid pathway to actually for the effector to bind and access this strained state. Okay. So to actually approach designing this, we started with CS221, which is a hinge protein that we previously had designed in the lab. It switches between one state, which we call state X, and a second state bound to an effector, which we call state Y. So it's this two conformation switch that looks like this. So you can see this domain here is kind of swinging back and forth in response to binding this effector. And then as a model heterodimer interaction, we chose LHD-101 here, which also was previously designed in the Baker lab. And then now we fuse these together such that the target can bind normally in this conformation, and we're building a little bit of additional structure here in gray to make that fusion work. But when we add the effector, or in the effector bound conformation, now we have this steric clash. So it was kind of a precise we like chose the geometry precisely so that here the target can bind normally. When the effector is bound, you get this clash. So these now should allosterically couple the target and effector. And so we made this design in the lab, and we measured. Let's see, we measured the association rate of the effector in various concentrations of target protein. So, and what we saw is as we increase target protein, so this is low target protein, this is high target protein, the rate of association of the effector gets slower and slower. So for one, this is great because it shows that we indeed designed this allosterically coupled complex, but also it's not really what we want because it shows there's not this rapid pathway for the effector to bind and displace the target. So now we move on to the second step. How do we design this like rapid pathway for effector association? Um, yeah, and so the reason that there's no rapid pathway for effector association in this case is with this hinge switch that we used, the binding site for the effector is actually closed when the target is present. And what has to happen is the target has to just kind of switch into this strain state, which is clashing with the target uh, first, then the effector can bind to stabilize that state. So in terms of energy landscapes, we might have this, nice strained ternary intermediate, but there's a really high energy barrier to actually access that, where this host protein has to switch into this really unfavored state, or state that's unfavored unless it's being stabilized by the effector. But before the effector is bound, that's slow to access. So instead, what we wanted to do is design a different looking state that the switch occupies when the target is bound before the effector is bound, which could, in theory, bind the effector. And so to do this, we started from this state Y of the complex, and we um, split the domains here and then translated this domain backwards by one heptad along the axis of this helix. So if you look, this is kind of the new conformational change we're trying to design. And the nice thing about this translation, rather than a hinge-like rotation, is that this preserves an open cleft where that effector could kind of weakly associate and induce the conformational change. So then we close this loop here, and we paired this state with the original effector-bound state Y of the switch. And now we do two-state design. So we use protein MPNN to choose or to simultaneously like optimize single sequences for both of these conformations. So so we're doing multi-state design here. Now, in this state, the effector can, or the target can bind normally, but we do have this open cleft where if we add the effector, it can kind of weakly bind, induce this conformational change, which because it's clashing with the target here, causes some, yeah, I should mention this is not like a physical of like a real state. I'm just showing that that this clash to show the frustration in these molecules. What really happens is that the protein complex will deform quite a bit when this switches into state Y. That puts a lot of strain on this interface with the target, leading to its rapid dissociation. Okay, so that was how I approached the design. Now, let's see, do those actually work? So to test that, we did an SPR experiment where we have the targets captured on the chip. We flow over the host, and that will bind. And then that's going to dissociate spontaneously at some rate. We want to see if we add effector, can we see this switch into a ternary complex and then dissociate much faster? And so when we actually do this experiment, we indeed see that the base rate of dissociation of the target is quite low. But as we increase the effector concentration, that dissociation rate gets faster and faster, which is exactly what we hoped for. It was a really exciting day when we got this result. Um, and if we plot that target off rate constant versus the effector concentration, we see this nice hyperbolic response where it starts off linear and then eventually uh, starts saturating at some maximum value. And if we actually measure directly the dissociation rate of the strained ternary complex, so from here to here, that is exactly the rate that this hyperbolic approaches, which strongly suggests that that ternary complex is indeed an intermediate in the overall facilitated dissociation process. Okay, so we wanted to explore a little bit more the actual mechanism of rapid effector association to form that ternary complex, because that really seemed to be the most important feature of this overall process. And when we did these experiments, we used this flexible effector, which actually, if we look at a CD here in orange, is mostly unfolded in isolation. We wanted to then compare that to the behavior of a rigid effector, which has these additional two helices to make this into a folded protein, even in isolation, though it binds with the exact same interface. So when we do this facilitated dissociation experiment with a rigid effector, what we see is we do get an effector concentration dependent increase in target dissociation rate, but we saturate at this much lower level. And if we directly measure the dissociation rate of the target from the strained ternary complex with the rigid effector, so this step, that level is actually up here. So this suggests there's, in contrast to with the flexible effector, there's some concentration independent rate limiting step that only is present with the rigid effector. So we wanted to figure out what that could be. Another clue came from doing association rate measurements of the effector to form the ternary complex. So with the flexible effector, we see, as you'd normally expect, a linear dependence of the on rate on the effector concentration. But with the rigid effector, we see a similar hyperbolic dependence as before. And we're again saturating at this low level, which is basically at the same level as here, the overall facilitated dissociation experiment. So the simplest interpretation of those results is that with this rigid
Adam.
effector, oh yes,
Yes, just a question here. I'm trying to like not lose the flow,
yeah.
and I might take you a little bit back in order to understand the core. So we have this the target host effector, the THE, and I'm trying to understand. We have the effector, especially the effector, and I want to think about it as, and I could be wrong, you will correct me, as a variant of a protein, maybe a peptide, like I'm seeing here. Now, the question is, before we look at the effector, what is the problem? Because I'm looking at an effector as some sort of a catalyst, and I could have misunderstood this still, to push this complex into the next state, probably.
Mm.
Now, my question is, without the effector, what is the problem with the target and the host, and what are we bringing by bringing the effector into this process?
Oh yeah. So the target and host have so what I really wanted to design here is uh, a system where the target and host have high affinity, and so they will dissociate very slow. But I also wanted a way to add a third component, the effector, so that I could accelerate the exchange, the dissociation of the target. So you could have both a high affinity interaction that you can also rapidly turn off.
Okay, okay, makes sense. So you want a high affinity, but also when you want to dissociate, you want it to be rapid, like fast.
Exactly.
Oh,
Exactly.
fantastic.
Okay, so let's see. So um, the interpretation of all of these results is that the rigid effector uh, is rate limited by some conformational switching before the effector binds. So first, the system has to switch into this strained state Y, and then the effector can bind to stabilize that state. And then once you get to the ternary complex state, then the target dissociation is quite fast. But in contrast, the flexible effector has to is actually able to directly bind to the state X, induce the change to state Y, which puts that strain on the target and leading to rapid target dissociation. So thinking about this a little bit almost like provocatively as in terms of protein motors, the idea is with uh, the binding of this rigid effector is kind of like a ratchet where you have to switch all the way into this really like highly strained uh, unfavorable state, and only then can the rigid effector bind to stabilize that state. So you have this really high um, energy barrier for the whole process. Whereas in contrast, this flexible effector probably is able to make interactions in this cleft and stabilize small steps along the conformational change coordinate, something like this, uh, so that by splitting this uh, large step into a lot of small steps, each of which can be like individually stabilized and so has low barriers, overall you can get a much more rapid induced process, much like a power stroke in motor proteins. So, and this is some new data that we haven't published yet. We want to explore a little bit more how this induced folding of the peptide is actually able to generate force. So the idea is that we take this peptide in the unfolded conformation and we tether it between 2 points in an atomic force microscope. Then the idea is when we add in this switch, it should induce the folding of this peptide and generate a force on the two ends here, this contractile force. And this is just some like back of the envelope calculations, but es essentially we know that binding the effector can release at least 5 kcals per mole of energy, and that change in distance is about 3 nanometers upon folding. So that works out to about maybe a 10 piconewton force that effector folding could generate. So we can do this experiment. So the setup here is that our collaborators have the effector bound to a target, and then they're kind of actually doing the reverse. So they're pulling on the two ends of the effector and watching it unfold, and then refolding will be the next thing that we do. And so here in purple, what we see is that as we extend and pull on that complex, the force goes up, and then eventually at 66 piconewtons, there's a jump or a decrease, and that corresponds to unfolding of the effector. Uh, and so yeah, so we can resolve this, and that's really cool. And the next step is we'll instead of fully unfolding the whole thing, we'll instead only go up to maybe here, and then slowly go back down. And what we should see is that we would hope to see a jump around here again, corresponding to refolding of the effector, and then we can directly measure how much force that generates. We've also done this, we've done this a few more times and looked more carefully at what happens after the effector's unfolded. And uh, curiously, we see a lot of these like intermediate states for a lot of these different traces. So like here, there's some like slow variation here. All of this is suggesting that even in the unfolded state, that effector is still able to make interactions um, with the host protein. And uh, kind of further confirming or pointing to that this like induced fit, induced folding method or mechanism that we're positing here. Okay, so I showed that
Adam,
yes.
there is a question on that previous slide.
Oh yeah.
Um, yes, so this industry switch we are talking about, the sort of like power stroke, I think, that you talked about. Is it the one that is, sorry, is it the one that is inducing this folding, is responsible for this industry folding?
Yes, yes. Yes, that's the idea. This is kind of new data. We haven't totally fully worked out everything here, but what we think is that this these like transient interactions that we're seeing after the main unfolding event is pointing to the effector being able to interact with the host even in its unfolded state, which we know or we think at least we has to be happening from the kinetic data that we have. This is more direct confirmation of that.
Okay. Then like, there is also a previous concept you've talked about, story, I think. So, do all these two processes like aim for the same thing? And if so, what makes them different?
They're all studying the same system, um, and just trying to probe different aspects of this overall mechanism that we've designed.
Okay.
Uh, so so the kinetic data, we can measure binding kinetics on like the overall process of facilitated dissociation. We can also measure the kinetics of certain individual steps in that process. And then here, this is less measuring kinetics, it's more measuring uh, interactions and how those are able to like, yeah, generate these forces. Uh, yeah, I don't know. I don't know if I'm answering your question very well. Is,
Yeah.
does that help?
Yeah. Yeah. A little bit.
Okay,
It helps. Yes.
cool. Okay. All right. So I'm moving on a little bit actually from kinetic data. So the kinetic data like showed that indeed I was able to design the function that I was interested in, which namely induced acceleration of dissociation. But I also wanted to show that the structure or like the proteins that I designed are working for the structural reason that I designed them to. So to get at that question, we solved crystal structures of the four like major states that these systems populate. So first, we solve structures of the apostate of the host and the effector bound state, and these indeed show the design conformational change, where in response to binding the effector, this domain here slides to the left in when the effector is bound. We also solve structures with the um, target bound, and then both with and without effector. In the target bound structure, pretty much agrees very nicely with the design model. And then the really cool thing is in the structure of the ternary complex. Here in colors, I'm showing the design models, but where you have this really strong and non-physical clash. The crystal structure shows exactly how the strain in this overall molecule caused by the clash resolves. And so you get this like quite significant bending of the target relative to this side of the complex. One thing that was curious in um, when comparing these structures is so here in gray is that ternary structure, and then in colors are the two binary structures. And I'm overlaying the two different interfaces we have. In the interface with the target is uh, is quite deformed in the ternary complex compared to in the binary complex. In contrast, the interface with the effector is not really seemingly affected at all. This point will become a little bit more important later, so just kind of keep this in mind. The target in these designs seems to deform more significantly than the effector. Okay, so um, from that kinetic data that I showed a little bit before, we saw, some quite significant enhancement of target dissociation. And the maximum rate of target dissociation is maybe around 0.2 per second. However, we saw that the rate of effector association quite outstripped that. It could get much faster and really showed no signs of slowing down. So the thinking was that we could probably modulate this and make the induced acceleration or the fold acceleration of target dissociation quite a bit higher if we were to change this energy landscape a little bit. So because there's seemingly like more room, like you can get the effector on really fast, and so we should make then if we were to increase the energy overall, the strain energy in the ternary complex, so raise this energy level, that could maybe raise the energy barrier for effector association, but not so much that it would become the rate limiting step. Uh, like there's room to raise this energy barrier while still shrinking this energy barrier so that overall the rate through this entire process would be faster. So to modulate the strain in these complexes, we sought to rebuild the fusion here between the switch and the binder to the target so
Adam,
that, yes.
I'm asking if you could, how can I think about the strain? Like, what mental model could you give to someone in this context? Like the strain within this complex?
Yeah, so you're designing these systems which um, you have some sort of clash there. And that clash is not possible in the real, like, protein structures can't overlap. So what's going to happen is the entire protein is going to bend and deform and strain somehow to resolve that clash. Now, that's great but that's going to come with some like energetic penalty because it's not the most preferred structure for these proteins to adopt. And so on the one, like, so that's exactly kind of what we're using. We're using the energy of that strain to break some interactions with the target and make it a much weaker affinity so that it dissociates quickly. So we're using that fact, but also it makes it hard to access that state because you have to kind of pay the energetic cost of forming that strain state. Um, So here what I'm saying is that the effector, because its association rate can get quite high, even though it's forming this strain state, it seems like we're kind of overpaying that cost, if you will. Um, so we could get even more strain than we currently have by
Okay,
binding this effector.
makes sense.
Yeah. Yeah, I'm sorry, I don't think I explained that point very well. So, um,
It was great.
yeah, so then the idea is if we raise the energy of the strain ternary complex, that will raise the barrier for effector association. So we're making more use of the overall energy that we get from binding the effector. And because then the energy of the strain state is higher, that should reduce the energy barrier for target dissociation, leading to overall a faster facilitated dissociation process. Okay, so to actually design this, we sought to explore different positions of the switch relative to the binder to modulate both the size and also the direction so the size of the clash and also the direction of deformation in response to that clash. So to do that, we sampled relative positions of the switch relative to the binder and rebuilt these three helices to stabilize those new like fusion positions. And so this is what so we have a whole bunch of different we made a whole bunch of different designs with different like directions that they would push on the target. And when we test all of these, we see quite a range of kinetic behavior. For the most part, they don't work very well, but there's a few designs, particularly these three, which show quite dramatic accelerations of dissociation in response to the effector. So this was quite cool to see. This one gets up to maybe 2000 or 2500 fold acceleration. So I wanted to highlight two of these designs as kind of exemplars of more general behavior that I noticed when comparing all of these around 40 designs. So this design, AS117, was the fastest one. This is the one that showed both the highest uh, accelerated dissociation rate and also the highest total acceleration of dissociation. Uh, And this design is the one that had the largest predicted deformation in response to binding the effector. So it was already curious that those weren't the same. Uh, this design, despite having about half the size of the deformation as this one was like 40-fold faster. And I think that the difference is that this one, um, the predicted deformation is downward, whereas this one, the predicted deformation is more outward. So if we look at these deformed states, if I cycle between them a little bit, you can see this one is bending downwards, and this one's predicted to bend more outwards, like that. And then and then looking at kinetic data here, we see that yeah, the accelerated dissociation with this design is about 40-fold higher than with this one. So what's going on here? Why is it not the case that the design with the biggest predicted deformation also has the fastest accelerated dissociation kinetics? Um, and I think really the reason for that is that the direction of deformation is important. So in this case, because it's deforming downwards, that's kind of that's parallel to or that's in the direction of like shear for these beta sheets. And beta sheets are really stiff against shear. So you could deform downwards just a little bit in the shear direction for a beta sheet, and that should be a quite high energy deformation. Whereas this one, where it's deforming outwards, you can just bend the beta sheet. That maybe is less destabilizing, or you have to bend a lot further around a beta sheet to actually get the same um, energy of deformation as you would in the shear direction. So this is also something that it's kind of hard to get at these questions directly here, but this is what we're thinking is that proteins just have, depending on the direction in which they deform, there are different stiffnesses associated with those directions, and therefore the same magnitude of deformation will lead to different overall energies of those complexes. So to explore this a little bit more,
I have a question here.
yeah,
Yes, it's direction should matter, I think, when it comes to protein molecules and so on, since most of the interaction and the I mean the conformational shapes of these protein molecules depend majorly on hydrogen bonding. So yes, I would expect that to differ when you change direction of the molecules in the way that they are formed. But the question is actually not directly going to be for this protein, but how do you factor in this machine learning general things? How does one consider uh, the energy of bonding or the energy within all kinds of energy that maintains the shapes of these molecules. It might not only be for these peptides or the proteins or another biological uh, form where you have to consider some bit of energy ideas here and there. So which energy kind is it the hydrogen bonding energy that comes in? Is it the some of the covalent bonding? For example, in proteins, you'll have the disulfide bonds that keeps them in shape, and then the hydrogen bonding. And then also you'll go ahead when it's in solution form, there is the hydrophobic status that keeps that determines which part of the protein faces out, which one faces. So which energy forms are usually considered in this machine learning or in this kind of design usually?
Yeah. So that's exactly right. There's all these different contributions to how or energetic contributions to protein folding. Back in the old days when we would use tools like Rosetta, which um, basically they just have an energy function with a whole bunch of different contributions, including all of the ones that you described. Um, and then the way we do protein design with a tool like Rosetta is we do Monte Carlo sampling of protein structure and also sequence to try to minimize the energy function. And if we can get to proteins that or designs that have low enough um, predicted energies, then maybe that would be a stable enough structure that it would actually form. So there, I think like the most important contributor generally to protein folding is the hydrophobic effect that you mentioned, where uh, hydrophobic cores are shielded from water and polar charge residues are presented to the surface. Uh, and then hydrogen bonding, of course, also particularly for forming secondary structures like helices and beta
Yes.
sheets. Uh, and then so that was kind of the old days. In modern times, we don't really have to explicitly consider that anymore because the machine learning tools have kind of have learned enough patterns about how proteins structure and sequence forms that it kind of automatically uh, like chooses like RF diffusion, for
Oh,
example, positions secondary structures. Yeah, go ahead.
I think I would also think that it will be since you're going to put in the sequence in there. So you're going to have the amino acid sequence, and there could be some algorithm that this machine learning follows to know that if it's amino acid B, if it's amino acid C, this amino acid is acidic, this is has hydrophobic chain, R chain, side chain. I think should be using the status of those amino acids in the sequence to determine the kind of energy or the level of energy associated, or how many hydrogen bonding can this form with another molecule, or will this one be facing outwards in when it's in solution form? Maybe it's something of that sort. But since I don't have a very good background of machine learning. Yeah, I'm trying to figure that out now. But I have very good background of the molecular component and the cell biology bit. So I would think it's going to be that way.
Yeah, no, I
Maybe
think
the
that's exactly right. Yeah. Um, so tools like protein MPNN, for example, will uh, rather than doing energy-based sampling, they do it's like a probability-based sampling, which has a lot of uh, connections to energy-based sampling, like Boltz, like probability and energy are connected via Boltzmann distributions. Um, and what protein MPNN is particularly looking to do is to for a given structure, maximize the probability of sequence. So it's finding the sequences that will most probably fold into the given structure. And it does that by looking at the kind of surrounding chemical environment for each residue, for each amino acid, and then choosing or like computing the probability of each amino acid given its local environment, and then choosing and then sampling from that probability distribution at each position.
Okay, thank you.
Yeah, I would think if you want to learn a bit about that, I think that paper is great to read. It was Eustace Dauperis in 2022. Okay. Oh, I have to speed up here. Okay. Okay, in that case, I'm going to skip some of this stuff. And then I want to now switch gears to talking about, so like, I told you how we kind of made this function where we could induce the acceleration of a dissociation in a protein. Now I want to show some ways that could actually be useful for or have some like biological applications. So so in particular, uh, I was kind of interested in whether this could be used to control cells in new ways. And the system I looked at particularly was IL-2. So IL-2 is a central immune cytokine, which generally activates T cells. So it tells them grow, divide, attack things, or regulate the immune system, that sort of thing. And it does this by dimerizing on the cell surface of immune cells. It dimerizes the IL-2 beta receptor and also the common gamma receptor chains. So when it does this, it turns signaling on. But natural pathways for turning this off are slow. So like you could imagine, for example, adding an antibody which would compete with IL-2 binding, but you first have to wait for this very slow process of spontaneous dissociation of this cytokine receptor complex before that antibody is able to stabilize the disassembly of that complex, just like I was showing before. The other way is that for these complexes to be turned off is that they'll eventually be internalized and degraded, but overall, both of these are slow. These would both happen maybe on the time scale of like at least 30 minutes to maybe more like hours. And we really wanted to find a way to turn signaling off within seconds. So to do this, I wanted to make a switchable version of IL-2, which much like the previous designs I showed, have this second binding site for the effector, so that the effector could bind, induce a conformational change, which breaks the interaction with one of the receptor chains, leading to dissociation of this receptor complex. Okay, so to actually achieve this, I started with a de novo designed mimic of IL-2 called Neo2, and I used that same switch that I showed before. And then I fuse them together so that when the effector is bound, the switch clashes with one of the receptor chains, but in the absence of the effector, that receptor can bind normally. So the idea is that if you add this effector, it binds, induces this conformational change, leading to receptor dissociation. Okay, so we did similar SPR experiments as before with that receptor chain down on the chip. We pre-associated the cytokine receptor complex, and then we flowed over effector and watched that cytokine receptor chain dissociate. And what we see is that at low or at no effector concentration, that dissociation rate is very low, but as we increase effector concentration, it gets faster and faster. And again, we see a similar hyperbolic response to increasing effector concentration on the receptor off rate. And then our collaborators did one of the coolest experiments ever, which is they actually could measure this process on the surface of live cells. So to do this, they use single molecule microscopy, and then they fluorescently label those two receptor chains. And then they're tracking colocalized pairs. They add the effector and then watch as a few seconds later that complex dissociates. And so then here I'm showing tracking for many receptor pairs. And if in blue, if we don't add the effector, nothing really changes. Those complexes remain dimerized. In orange, if we add the effector at time 0, within about 10 seconds, all of the complexes have just completely dissociated. This worked like way better than I ever would have imagined it to. Um, and then this also has a profound effect on signaling. So here I'm showing STAT5 signaling, which is uh, the primary output of IL-2 signaling. And then starting at time minus 5 minutes, at time 0, I either add nothing, and then we just in blue measure a normal signaling time course. In orange, I add the effector, and what we see is that signaling immediately stops accumulating and then instead slowly reverts to this low level. And it's just about as effective as ruxolitinib in green, which is an inhibitor of a key kinase along the signaling pathway. Uh, so, but that's like a small molecule and irreversibly modifies the cell. So the effector is a way to have the same effect as this small molecule inhibitor, but without actually modifying the cell at all. Okay, and then finally, I wanted to see how transient stimulation with this tool can actually affect cell behavior. So here, so the idea it would be you can transiently stimulate cells using this switchable cytokine by first adding the cytokine that turns signaling on, and then maybe a few minutes later, you can add effector and that should turn signaling back off, even if that signaling has already started. And what we see is that this can decouple certain functions that maybe occur really early in signaling from others which require more sustained stimulation. So here we're measuring, we stimulated cells and then measured them 3 days later. And what we see is that compared to unstimulated cells, ones that were stimulated for even 5 minutes are surviving at about double the rate of unstimulated cells. So even a short transient stimulation is able to activate like some anti-apoptotic programs, something like that. However, cells that were not stimulated or that were transiently stimulated are not dividing any more than the unstimulated cells. So really, it seems sustained stimulation is required to induce and drive progression through the cell cycle. So I thought this was pretty cool. You can uncouple these fast early responses from later ones in IL-2 signaling. Okay, and then I have a whole bunch of people, mostly from the Baker Lab, to thank. My two mentors, David Baker and also Florian Praetorius, and then everybody else that I worked with. And then also my collaborators. We had several collaborators for this project, and they could all perform these amazing detailed experiments that we don't have the instrumentation for. And my committee and also IPD management. And yeah, happy to take some more questions.
Uh, I felt like maybe it is answered at the end, but maybe you'll clarify. I'm wondering like if I wanted to slow down the dissociation intentionally, like which parameters should I look at or adjust? But you talked about induced stimulation, and I was like, maybe that would. But go ahead. Where should I look?
So if you want to, sorry, if you want to slow down dissociation and start from a very low dissociation rate.
Okay. Yeah, like I wanted to slow down dissociation intentionally.
Mhm.
Like, are there parameters that I should adjust within the process?
I see. Uh, yeah, so this is honestly, this is something that is one of the biggest challenges for designing other facilitated dissociation systems, because not all interactions that you start with have low dissociation rates that can be dramatically accelerated. Um, and oftentimes that's a feature that you really like want. So I think the best way to get, or the most reliable way, I guess, to get slower dissociation is to design bigger interfaces that make more contacts. Um, Beyond that, uh, yeah, I think it's just like a lot of like sampling of binders to a target, and then we still don't really know what makes one binder like have a picomolar affinity and the other binder have a micromolar affinity. So yeah, that's still a super hot area of research. I don't know if I'm quite answering your question though, or if you meant something else.
Yeah, yeah, sure, you are. I think I don't know if there is anyone in the audience with a
I,
question in
yes,
the
I have another question, a brief one.
sorry.
This is something I keep asking myself. What are the chances of in silico designed proteins being functional in a natural state? Let's say you have designed a protein, then you're able to synthesize the gene for it and put it maybe in put it in some uh, external, you put it in some bacteria or some fungus and grow it up.
Mhm.
What are the chances of these proteins being able to get um, synthesized by these bacteria, and also checking that whether the conformation will match with what is in or what has been designed in silico?
Yeah. So this kind of depends. This depends a lot on the specific structures you're trying to design and the function you're trying to design. But just like in my experience, I'd say like over 90% of the proteins uh that I that I design do seem to express like maybe even like express really well and be like stable as monomers. And so they don't form aggregates or anything like that. Maybe there's like an 80% success rate for being
Yeah,
expressing
that,
and being like stable. And then
I think for biological thing, 80% is good enough. But then if you
yeah,
have a complex protein, let's say where two peptides have to associate and so on, that will won't that put in some more challenges? I don't know if that
oh
have
yeah,
actually been designed and you expect it to work out natural state because of the complexity of how the cells work.
oh, specifically this short little peptide, do you mean?
The little peptides should be easily
Yeah,
expressed, but in any case, if you to design, maybe you want two, let's say just two peptides, you want to design them and they should be able to complement, I mean, bond with each other and form a secondary structure, which should become
mhm.
functional. Have you ever come across any studies like that, or have you ever seen someone do that, attempting
Yeah,
something that
I have seen that. I don't we don't really do much with like th
wetland?
those kinds of structures are honestly a little like um
Yeah.
simple for most of the functions that we try to design. But people have used these like coiled coil uh heterodimers to do a lot of cool things. There's one group at a university in Slovenia, I think, called Roman Urala, who has done a lot of work with coiled coil, or so like two like interactions between two helical peptides, and he can use that for a lot of cool functions in synthetic biology.
Okay, thank you, man. Thank you so much.
Yeah, totally.
Adam, thank you, Adam. I think the last one for me is still about strain. Like, if you have more strain within this complex, is that good? Like, is it better? Or like, when does the effector, like the binding of the effector becomes sort of like a bottleneck within this strained complex?
Yeah, so I never reached that limit. And I think actually I'm more limited by the, I can maybe go back here a little bit. Or yeah, here's a good picture maybe. Well, I'll go back a little further. Oh, okay, yeah, something like here. So here, like I think I'm really limited by the size of the conformational change that I get. So um, the total amount of strain that I can get is maybe like, this much in the system. Just because I'm limited by the overall size of the conformational change here that's induced by the effector. So yeah, I never saw a case where effector binding was actually like well, unless it was a bad design. But yeah, I never saw a case where I was able to make effector binding the rate limiting step and be slower than target dissociation at a really high effector concentration.
All
Yeah.
right, all right. I think that brings the session to the end. I want to thank Adam for taking of time and really share this brilliant work with us. Thank you very much, Adam. We can't thank you enough. And big thanks to the members of the Computational Intelligence Group. Thank you very much for attending. And I think we'll see or we'll meet again in our subsequent sessions.